Hello, everyone. My name is Antonia Ramos, and I'm a student from the Faculty of Science of the University of Chile. And on this occasion, I'm going to present our research named Functional Zebrafish Microbiome as revealed by global proteomic profiling of control and infected larvae. The animal microbiome plays a key role on different physiological processes, and it also affects the whole cell. That's why it is important to study changes in the presence of pathogens. Here, we decided to study those changes using zebrafish larvae infected with Pseudomonas by using two different methods, injection and immersion. To analyze changes on the microbiome, instead of using a more classical approach such as genomics, we decided to use a proteomic one because it allows us to not only study the different taxa that are present, but it also because it gives us the possibility to understand the microbiome at a more functional level. And so zebrafish larvae were infected by two different methods that I already mentioned, injection and immersion. And then the peptides were extracted, they were also digested, and then they were analyzed by a high-resolution mass spectrometer. After that, the raw data that was provided by the mass spectrometer was mapped with the different possible peptides, and then those were mapped with the UniProt database to determine the corresponding proteins. And once we knew the proteins that were in the different samples, we were able to obtain a global proteomic profile to identify the taxa of each one of the proteins and also recognize the different functions in which the proteins were involved. After that, we obtained the following results. First of all, we can see that the percentages of bacteria peptides that were identified of the different conditions. And secondly, we can see the taxonomic diversity here. Here we can see a quite similar diversity in both control samples, but we can also see that in the case of infection by injection, the diversity dropped down drastically. But what is interesting here is that when they were infected by immersion, in this case, there's not a big change on the diversity of the taxas found. This may be explained by the fact that in the immersion case, the pathogen is not capable of actually colonize the larvae, and therefore it does not affect the diversity of the microbiome. This can also be observed on the functional level. Here, we can see there are no major changes on the peptides found in the control and in the infection by immersion samples. Whereas in the case of infection by injection, there are substantial changes on the peptides found in comparison with the control samples. Therefore, a functional change in the microbiome is observed when larvae are infected by injection with Pseudomonas. In conclusion, we can say that the proteomic analysis is a powerful tool for studying changes in the microbiome, in the changes in the zebrafish larva microbiome, because it doesn't only provide information regarding the diversity of taxas, but it also provides functional information of the microbiome. Finally, our study highlighted the importance of the infection method used, in this case, injection and immersion. Thank you so much for your attention.